All right, this is Chapter 11, Campbell's 7th edition biology on cell communication. And we'll run through this pretty quickly, not too complicated of a chapter. So we're talking about the ability of cells to talk with one another over short distances and long distances. So for example, on a simplified model here we have is yeast cells where they exchange mating factors. Each one secretes its own protein, but they bind to a receptor on the outside to exchange information or what have you. So they cause changes internally that triggers a fusion or a mating, if you like, so that an A cell and an alpha cell becomes an A alpha cell. So signal transduction pathways basically take signals from outside the cell that will bind to the cell surface and convert them to intercellular responses, or those occurring within the cells. And this exists in all different forms of life, from prokaryotes up to you and mammals, other mammals. So it therefore suggests that they're evolutionarily conserved, another form or basis for the argument towards evolution. So let's look at different types of chemical messengers. We already know that information can be passed from cell to cell. In animal cells, we have gap junctions, pores that go back and forth. They can be passed. And in plant cells, we have plasmodesmata, again, the pores through the cell wall and the membrane in this case, that can pass nutrients or proteins, whatever, back and forth. Sometimes cells will bind directly to each other through glycoproteins. This can be seen sometimes in the immune system, for example, when one cell binds to another to destroy it, such as a killer T cell, for example. But this is local signaling. You have one protein from cell surface binding to another protein on the cell surface and triggering a series of reactions inside the cell. There's also cases where one cell secretes stuff. So here we see paracrine signaling, which is nearby. This one cell releases just a bunch of products, and all the cells nearby in the area receive that chemical and cause changes internally. And here you see another type of local signaling here where we have a neuron releasing neurotransmitters into a synapse to then bind to the target cell, whether it's another neuron or a muscle or something like that. Then we get to long-distance signaling, which will be the focus of the rest of what we're talking about here, such as that in the endocrine system with hormones that are released from an endocrine cell and put into the bloodstream. And these hormones will travel through the bloodstream and bind to receptors or go into cells far away from the original area where they were released. Now, a lot of this originated from Earl Sutherland, who won the Nobel Prize when he discovered how the hormone epinephrine acts on cells. And he suggested that there was a series of steps that cells went through to receive and process the signals, and he called it reception, followed by transduction, and then eliciting a response. So reception, transduction, response. And you can see that here. This is linked to an animation, if you like, from the real PowerPoint, not from this one specifically. But here you see reception followed by transduction, where there's a relay system where one communicates to the other, and then finally you get your response, something happening, making a molecule, breaking it down, turning a gene on, that type of thing. We often call this molecule that binds to the membrane a ligand. And the ligand is very specific for the receptor that it's going to bind to. And typically it causes this receptor, which is a protein, to undergo a conformational change. And that conformational change triggers a series of reactions internally. Sometimes molecules go directly into the cell and bind to intracellular receptors that are cytoplasmic or in the nucleus. And these are typically hydrophobic and small signal molecules, and that enables them to pass through the membrane directly, so that because the interior of the membrane, which is nonpolar and hydrophobic, does not repel them. One example of this would be steroid hormones that bind to intracellular receptors. Steroid hormones are based on cholesterol. They're nonpolar. Here's an example showing you a steroid hormone, in this case testosterone, going through the membrane and binding to a receptor protein in the cytosol, which when bound, this is a transcription factor that turns on a gene and converts it to RNA and then a protein. So it triggers the production of a specific protein. Now, there's 3 types of receptors in the plasma membrane that we often see in many, many cells. G protein linked receptors, tyrosine kinase receptors, and ion channel receptors. So here's a G protein linked receptor. This is a ribbon model of what it looks like in the membrane you can see. Here's the filled model, if you like. Here's the G protein. Here, receptor. Here it binds to a G protein. You'll notice it's not active right now. It's just got GDP, so it doesn't have energy. It's inactive. GTP will bind to it and kick out GDP, activating or changing the shape of this protein, which will allow it to bind to the receptor that's caused by the signal molecule. And then once activated, this protein now will travel over and bind to this enzyme, which will activate it and cause it to have a conformational change and thus an internal cellular response, leaving behind GDP, and then we're back to square one. Tyrosine kinases are known for creating or causing phosphorylation cascades, where we have this dimer, these inactive monomers, which when signal molecules bind to those receptors, they fuse and dimerize, as we say. They form a dimer. These are called tyrosine kinases because sticking out of these monomers are tyrosine, which is an amino acid, 3 on each one. And to those tyrosines, we will bind 1 phosphate group each. So we're activating it by phosphorylating it, a total of 6 for the tyrosine dimer. And then each tyrosine that's phosphorylated binds to these relay proteins, these other enzymes, which change shapes and cause a series of cascades to cause some internal response. And again, these are generic. There's very specific examples, some shown later, and others that you'll look up as part of your animations. The last one here is ion channel receptors. You've largely seen this before. These are basically just allosterically regulated ion channels where here's the active site. Ligand binds, that triggers the gate to open, allows molecules to go through from high to low concentration, triggering an internal cellular response, of course, and when the ligand is removed, the gate closes and doesn't let anything else through. And we'll take a break on this one.